Thank you to the Caucus co-chairs and their legislative staff, Senator Wicker and Klobuchar, and Representatives Butterfield and Bilirakis. Also, thank you to the Every Life Foundation, especially Christina Hartman and Shannon Van Velden, for inviting me to moderate this session and for their hard work, really your hard work, in leading the legislative cause for patients with rare diseases. So thank you to the Every Life Foundation. And thank you to all of you for attending today and speaking with us about ensuring that patients with rare disease and their families are heard in our federal government. So today we have a terrific set of speakers who will update all of you on exciting new technologies and treatment opportunities for rare diseases. In highlighting these technologies, first let me just point to the development and use of genomic sequencing. Through genomic sequencing, we can examine all of a patient's genes in a single test to identify potential mutations that can lead to genetically based diseases, and thereby more rapidly get a definitive diagnosis, ending a patient's diagnostic odyssey. It's an important issue for patients with rare and undiagnosed disease. We can then share the sequencing data and outcomes data in global databases, linking the patients with rare diseases around the world, thereby having the ability to consider the treatment options tried in a number of patients with a disease, no matter how rare. And through that, we can really help understand what the best treatment course is and have that delivered to patients. This, in some cases, has led to significantly improved outcomes. Second, I'll point out the development of gene and cell-based therapies, an exciting new technology for which we'll spend most of this briefing talking about. These therapies can provide new therapeutic options that will provide significantly improved treatments, or in some cases, cures. They directly affect the molecular pathways that are affected in the rare disease. Gene therapies aim to alter the genetic mechanism that is the basis for a disease within a patient or their own cells, thereby restoring functional molecular pathways. This is an exciting new area. It's not exactly new, but it's just becoming available to patients recently. So it's new from a patient perspective to have this type of therapeutic strategy available. These technologies are all examples of personalized medicine, where we aim to determine the molecular characteristics of a patient, diagnose their disease as early in care as possible, and design and implement treatments that will target their disease based on their specific molecular characteristics. Personalized medicine, the catchword for it, is getting the right treatment to the right patient at the right time. I, as Christina mentioned, am Daryl Pritchard. I'm the Senior Vice President for Science Policy at the Personalized Medicine Coalition, an education and advocacy organization based right here in Washington, D.C. The coalition is a collection of many stakeholders in the field: biopharmaceutical companies, therapeutic companies, diagnostic companies, laboratories, healthcare delivery providers, both academic health centers and community hospital systems, healthcare delivery support, healthcare policy organizations, payers, and most importantly, patients. We all come together within the Personalized Medicine Coalition, the PMC, with the shared objective to advance personalized medicine. We work with the Every Life Foundation, as well as with members of Congress, to bring awareness of new technologies in rare disease and to advocate for appropriate policies that will lead to the access to these new technologies and ultimately to improved care. Today, we're going to welcome some of the country's, really the world's leading experts in biotechnologies to discuss breakthrough treatment options for patients with rare diseases. And we will talk with a sickle cell disease patient and advocate who has participated in a clinical trial for a new gene therapy and could describe her experience firsthand with this promising therapeutic strategy. Together, we hope to make the case clinically, economically, and interpersonally to support legislation and policies that will help advance these technologies so that more patients will have access to the best care and be put on the path towards potential cures. So joining me today are Doctors George Church, Stanley Nelson, and Gary Gibbons, as well as Tasha Samuels, who's from the Washington area and can bring a particularly potent and necessary perspective.